This is our genetics presentation on infantile Tay Sachs disease for our human genetics class. I'm Kaylee. We also have Ava, Jordan, Rodney, and Zoe. In this presentation, we are going to talk about the background of Tay Sachs disease, the carrier screening and testing that can be done, the importance, incidence, and etiology, the phenotypic features, molecular basis, pathogenesis or disorder, the inheritance pattern of this disorder, and the genetic principles.
Okay, hello everyone. My name is Ava. So first I'm going to talk about a little bit of background, and in order to do that, I'm going to talk about the case example that is in our textbook. So in our example, we are introduced to a couple initialed RT and ST, RT being male and ST being female. They are partners seeking counseling for possibly having children. They were referred to a genetics clinic because they're at risk of having a child with Tay Sachs disorder. Both of them are of Ashkenazi Jewish ancestry, which is really important because this ancestry is known to have very high Tay Sachs disease in their population. It's actually a 1 in 30 carrier frequency versus the normal 1 in 250 for like the general population. Along with this, ST reported having a sister that passed away in early childhood due to Tay Sachs disease. So this is a very important thing to consider when referring to their family history. And RT also reported having a paternal uncle who lived in a psychiatric home, but he did not say any specific diagnosis that the uncle has. So for our carrier screening and testing on the next slide, I drew a little pedigree. In the middle, you can see RT and ST. RT is the square indicating he is male, and ST is the circle indicating she is female. They also reported that they were informed as teenagers that they have the reduced hexosaminidase A activity. And this is important because they were thought to be Tay Sachs carriers because of this enzyme testing. However, they did go through with molecular testing, and this showed different results. So ST was confirmed to be a heterozygous carrier for the pathogenic hexa variant, as indicated with the darker circle inside of her circle. Tay Sachs is an autosomal recessive disorder. So this does mean that she does not have Tay Sachs disease, but she is putting a child at risk of becoming a carrier. RT did not have the disease-causing variant, but he did have a pseudodeficiency allele, which can make the enzyme testing appear abnormal, but he doesn't actually have Tay Sachs disease. So that's something also really important to note. And as well on the pedigree, you can see ST's sister drawn in with the completely shaded circle indicating that she does or did have Tay Sachs disease, and the line through it indicating that she has passed away. And in the upper left corner, you can also see RT's paternal uncle, who did have that unknown psychiatric illness as indicated by the shaded square. So on the next slide, I have some important things to talk about regarding this case example. The first one being the misleading enzyme testing. As I talked about before, doing the enzyme testing alone can be misleading because of a pseudodeficiency allele that can lower that measured enzyme activity in the lab. So this can make someone appear as if they are a carrier like RT, but they actually won't be at risk of having an affected child unless they have that true pathogenic variant that we talked about. So for this couple's results, As we talked about, ST is the pathogenic hexavariant carrier. So this does mean that their children would not be expected to have Tay Sachs disease, although some of them could be carriers. So in the bottom picture represented, as you can see, it does say that the carrier is male in this picture, although in our situation it is female, but it does show that their children will have a 50% chance of inheriting carrier status or a 50% chance of being non-carriers for Tay Sachs disease. So overall, this case really highlights the importance of testing, specifically combined testing with the enzyme testing and molecular testing, and also genetic counseling being a really useful source, especially for couples seeking out reproductive suggestions and guidance for possibly having children.
Hello, everyone. My name is Jordan, and I'm going to be talking about some of the phenotypic features seen in infantile Tay Sachs disease. So, in infantile Tay Sachs disease, phenotypic features can show up in a lot of different ways, but there are some common patterns that are usually seen. One of the first things people notice is progressive weakness. Babies may also become less attentive over time and seem less engaged with their surroundings. Along with that, developmental delay is very common, meaning they don't reach milestones like sitting up or moving forward when expected. Another symptom is an exaggerated startle response, where even small noises or sudden movements can cause a really strong reaction. You might also see hyperflexia, which basically means that the baby's reflexes are more active or stronger than normal. A key term to understand is generalized hypotonia with clonus. Generalized hypotonia means low muscle tone throughout the body. So the baby can appear kind of floppy or have trouble with their movements. When these happen together, it shows how much the nervous system is affected. In the picture shown, this is a baby with hypotonia, and as you can see, the adult is pulling up on the baby and their head is really drooped back, like they're not even giving any effort on trying to hold themselves up. And then when the adult has the baby facing downward, their whole body is kind of shifted towards the ground. They're not, once again, giving any effort to like fight it or tense up. They just have that really low muscle tone and aren't able to hold themselves up. And on the next slide, I'm going to be talking about treatment options. So even though there's currently no cure for infantile Tay Sachs disease, there are still treatment options that can extend life and manage symptoms. A big part of treatment is supportive, non-medication-based care. This includes protecting the airway to help with breathing, managing infections, and making sure the baby is getting enough nutrition, since feeding can be difficult. Seizure control is also very important because seizures can develop as the disease progresses. There are also some more advanced treatments being studied. One of these is enzyme replacement therapy. This involves giving the body the enzyme it's missing called hexa-A, or beta-hexosaminidase, so cells can function more normally. The enzyme is delivered into cells and sent to lysosomes where it is needed. Other options include hematopoietic stem cell transplantation, which aims to introduce healthy cells that can produce the missing enzyme. Another approach is substrate reduction therapy, which works by lowering the amount of substances that build up in the body because the enzyme isn't working properly. And lastly, gene therapy is developing treatment that focuses on fixing the root cause of the disease. It works by delivering a normal working version of the gene to replace a defective one. So overall, while these treatments don't cure the disease, they can help manage the symptoms and improve quality of life.
So I'm going to talk about the etiology and the incidence for Tay Sachs disease. So first, when we look at the etiology of the infantile Tay Sachs disease, it's important to honestly understand that the condition is an autosomal recessive genetic disorder, which means that a child must inherit 2 copies of the defective gene from each parent in order to develop the disease. The disorder is specifically caused by the mutation of the hexa gene, which is responsible for producing an enzyme called beta-hexosaminidase A. This enzyme plays a critical role in the body as it can help break down the fatty substances known as the GM2, and those are within your nerve cells. This enzyme is deficient. When the enzyme is deficient or absent, this is because of gene mutation. So the GM2 cannot properly break down things, and as a result, it accumulates in the brain and in the spinal cord, and over time that buildup becomes toxic and can lead to progressive damage of the nervous system. So infantile form of the Tay Sachs disease is the most severe and the most common type. The symptoms usually begin in early infancy, and then often within the first few months of life, the disease progresses really, really fast and leads to significant neurological decline. In terms of incidence, the Tay Sachs disease is considered a rare condition in general populations, occurring in approximately 1 in 100,000 to 320,000 live births. However, it is more common in certain populations, such as the Jewish population, where the incidence is much higher. It's about 1 in 3,500 births. So because Tay Sachs is inherited in an autosomal recessive pattern, if both parents are carriers of the mutated gene, there is about a 25% chance with each pregnancy that their child could be affected by this disease. So in this next slide, you can see on the left for the etiology, it kind of breaks down within the spinal cord and the brain cells where the damage happens, and then also the abilities that the baby or the infant does start to lose, and then the age. And then for the incidence, you can see how rare it is for just common normal populations on how small it is. So this is a pretty rare disease.
Hi, excuse me. Hi, I'm Rodney, and I'm going to be talking about the molecular basis in addition to the pathogenesis. As she mentioned, Tay Sachs disease is caused by mutations in the hexa gene. This enzyme is found in lysosomes and is responsible for breaking down GM2. GM2 is a fatty substance normally present in the nerve cells. And in healthy individuals, this process helps maintain normal cellular function and prevent buildup of waste materials. When mutation occurs in the hexa gene, as GM2 cannot properly be degraded, and then it begins to accumulate within the lysosomes of the neuron. And this accumulation is progressively and directly linked to the severity of the disease. Over time, the GM2 disrupts normal cell processes by enlarging lysosomes and interfering with cellular function. This molecule defect is the normal underlying cause of neural damage and explains why the disease primarily affects the nervous system. On my next slide, I also mention pathogenesis a little bit. The pathogenesis of Tay Sachs begins with the accumulation of GM2 inside the neurons, and when these lipids build up, lysosomes swell and interfere with the normal internal structure of the cell. This disruption is especially harmful in neurons, which are highly sensitive and depend on the process in the intracellular processes. Eventually, the damaged neurons will undergo cell death, resulting in widespread neurodegeneration. And this process is like a progressive loss of the neurons, and it's responsible for severe symptoms like blindness, seizures, and paralysis. Lastly, on my last slide, I will be talking about the pattern. As you can see in this pattern, when both parents are carriers, there's a predictable pattern of inheritance. About 25% chance, there's a 25% chance the child will be affected, a 50% chance that the child will be the carrier, and a 25% chance that the child will inherit two separate DNAs of the gene. And these probabilities are the same for all pregnancies. This inheritance pattern is important for implications with the family because this genetic will often encourage them to make reproductive smart decisions and get checked out, things like that. Thank you.
I'm Zoe, and I'm going to be talking about the genetic principles, or in this case, a little bit more what sets Tay Sachs infantile apart from other neurological disorders. First, we're going to talk about the eye findings. So typically children and those affected with Tay Sachs have a cherry red spot in their retina. This is due to a mixture of pallor due to the swelling of retinal ganglion cells and also the attenuation of the macula and the choroid. If you look at pictures, it's pretty cool. And then vision effects due to the mutations in the hexagene will lead to reduced vision, and progressive vision loss will eventually lead to blindness, and it can also cause the narrowing of the vision and the narrowing of the retinas. It's typically prevalent in the Jewish communities, Cajun communities, and in Quebec. That's where it's got the higher rates. And then neurological symptoms in infants versus juveniles. In infants, it's a little bit easier to tell. They become increasingly unresponsive and they won't develop as quickly. They won't be able to talk, or they'll develop and then it'll quickly decline. And typically seizures will begin around 12 months of age. And then in juveniles, they'll just start to have some dysphagia. So they'll have inability to swallow. They'll have some ataxia. They'll have the loss of coordination. So their ability to walk and their ability to do things with their hands and their limbs is going to go down as well. And they'll also become clumsier and weaker due to both of those things. And a vegetative state is typically reached by ages 10 to 15 years old. And it's very rare to have an adult with Tay Sachs disease. And that's it.
We've been waiting for so long.